Hey everyone, welcome back and thank you for your patience. We are going to cover the second part of the pulmonary system lecture, and in this lecture we're going to take a look at ventilation in more detail, including what drives ventilation. In other words, how do we actually inspire and expire? And then from there we'll take a look at the role of the pleural membranes in ventilation. And then we'll take a look at a couple kind of tangential case studies, including a pneumothorax, which is more commonly known as a punctured lung. We'll look at asthma, and then we're going to take a look at surfactant secreting cells, and then tie that into the disease process of COVID-19. It'll be a timely lecture. All right, so let's get started with ventilation. So ventilation is driven by a physics principle known as Boyles Law. You are not required to know that term, nor are you required to do any sort of mathematical calculation. However, you are expected to understand the basic principle of what I'm about to describe, or Boyles Law. Consider air. There's air here. What is all of this air? Well, this air is actually gas molecules that are running around. And if I take a box and seal a certain amount of air in that box, there are gas molecules that are contained, a set number, whatever I captured and sealed in that box. So those gas molecules are running around. Well, what are those gas molecules? O2, N2, a little bit of CO2, it's all going to be contained. And they're bouncing off each other and bouncing off the walls of the container. And when things bounce off the walls of a container, that exerts pressure. So there's a set number of gas molecules, and then they're bouncing off the walls, exerting a certain amount of pressure. So what if I take that compartment and I expand it while still keeping it sealed, and therefore the number of gas molecules is constant? So I'm going to expand the volume of the container, but the number of gas molecules is the same. Well, suddenly all those gas molecules have a lot more room to run around. They're not bouncing off each other as much. They're not bouncing off the walls as much. So they do not exert as much pressure. The converse is also true. If I take that box and I constrict it, then I'm basically crowding all of those gas molecules in this smaller area, and they start bouncing off the walls even more, and therefore they exert more pressure. So if you have a set or a finite and constant number of gas molecules in a container, and then you change the volume of the container, you change the pressure of the gas molecules. And I think it's better displayed in this image here. And if you look carefully at this image, you can actually count the number of gas molecules. The number of gas molecules is consistent here, here, and here. And what we've done is we've just changed the volume of the container. And you can see it's a rather linear relationship. If I double the volume of the container, my pressure drops by half. If I halve the volume of my container, or make it half as big as it was, then my pressure doubles. Okay, so why are we even exploring this? Just like fluid, air moves down its pressure gradient. So we actually see this with weather in the atmosphere all the time. When you have a high pressure system and a low pressure system kind of coming close together, it creates a lot of wind. In fact, that's one of the things that drives tornadoes. So air moves from high pressure to low pressure. So if we are going to move air in and out of our lungs, then we need to set up pressure gradients. If I want air to move out of my lungs, then the pressure in my lungs needs to be greater than the atmosphere, and the converse is also true. If I want air to move into my lungs, then I actually need to make the pressure in my lungs lower than what's out here in the atmosphere, and that's going to draw air in. So how do we create pressure gradients? Well, we change the pressure of some system. Well, which system are we changing? Well, out here is pretty much constant. So if you're at sea level, for instance, the pressure of air is 760 millimeters of mercury. That'll be a number that we'll revisit later on in one of the Zoom lectures. But I can actually change the pressure of air in my lungs by changing the volume of my lung airspace. So This is where the importance of having that rib cage, your diaphragm, your respiratory muscles like the intercostals, all comes to play. So remember that your lungs are basically going to be attached to the rib cage via pleural membranes. And so if I expand out my rib cage, I basically expand out my lungs, and that expands the volume. So what is that going to do to the pressure in my lungs? Well, if we look here at this diagram, by expanding the volume, the pressure drops, right? So we're going to expand the lungs, push down on the diaphragm, make our lungs bigger. The pressure is going to drop, and that is going to create a pressure gradient where now the pressure out here is greater than the pressure in here, and air is going to move in. So by expanding the volume of my lungs, that drives inspiration. For expiration, I'm going to let my lungs volume decrease. I'm going to let my rib cage relax, my diaphragm come up. That's going to decrease the volume of my lungs. By decreasing the volume of my lungs, I increase the pressure. Now the pressure gradient is such that the pressure of air in my lungs is greater than out in the atmosphere, so air moves out. So that drives expiration. This image here demonstrates that. So you can see as you follow along in this image that to drive inspiration, we're going to expand out the rib cage and push down on the diaphragm. What that's going to do is expand out the rib cage, and you can see in these arrows that's going to draw air in because the pressure inside the lung airspace is less. Conversely, if I want to push air out, I need to shrink the volume of my lung airspace. And the next graph is something that you may want to spend a little bit more time looking at than what I'm going to go over. It has to do with where we are in the breathing pattern. So what I'd like you to do for a moment is sit and just breathe normally. So when you breathe normally and you draw air in and then you push air out and draw air in and push air out, again, remember it's your rib cage and your diaphragm that are basically dictating is air coming in or coming out. But we can actually follow the tracing of air movement in and out of our lungs using something called spirometry. And so on this graph here, I'm going to try drawing it out for you a little bit. We'll see how this goes. So spirometry does something like this. And this is part of it here. What it says, okay, breathe in. So what happens? Well, air starts at a certain amount. Obviously, when you breathe in, you add more air, right? Like so. Okay, now breathe out. Okay, so when you do that, the air is going to go back out. Now, it's not that you have zero air left in your lungs. It's just that you're looking at, well, what are you moving? Breathe back in, etc. Breathe back out, etc. And this is sometimes referred to as tidal volume when you're just breathing at rest. It is covered briefly in Lab 18 and in much more detail in Lab 19. So if the term is a little weird at the moment, hopefully after those labs it'll be more familiar to you. So all I'm doing with this graph is basically showing air moves in, air comes out, air moves in, air goes out. It's basically looking at the volume of air in my lungs at any given time. So inspiration, expiration, inspiration, expiration, etc. Let's take a look at how this maps to the pressure in my lungs. So this image up here is showing you the pressure in the lungs. And what I want to do is just look at this little bit here. You can see the pressure starts at something, and then it drops, and then it goes back to the starting point. And then somehow that drives inspiration. Okay, so we're going to start here with our pressure. We don't know what number that is, it's just some pressure. Now what we're going to do is we're going to expand our lungs. So as we expand our lungs, the pressure is going to drop, as you see here. So well, what is that going to do? Well, that's going to create that pressure gradient that's going to start drawing the air in. So now the air is being drawn in. Well, as the air comes in, the pressure begins to equalize. So you expand the volume and then you stop, but the air keeps coming in and makes the pressure go back to its normal or starting point because again, you expand the volume and air gets drawn in until you reach pressure equilibrium again, at which point no more air moves. So you guys can give this a shot. What I would like you to try to do at home is take a deep but slow breath. So you're going to go and then hold it for just, 5 seconds. And so you've expanded your rib cage, you've drawn air in, and then what? Well, while you're holding your breath, the pressure in your lungs now is equal to the pressure out here, because if it wasn't, you would still be drawing air in. So again, you expand out your rib cage, pressure initially drops, starts sucking air in, but as that air comes in, you bring the pressure back up until it reaches equilibrium. So the converse is also true. So what we just saw, I'm going to box briefly. This is what we looked at. We're going to ignore, we're going to ignore the blue line. And now let's take a look at this part here. So you've reached the peak of your inspiration. You hold your breath, and now you're going to relax your diaphragm, let your rib cage contract. That's going to increase the pressure by decreasing the volume. So there's that increase in pressure. Now there's a pressure gradient. Air starts moving out, but as air moves out, the pressure is going to eventually reach equilibrium with the atmosphere. So if I were to ask you, at which point do we have maximum pressure in our lungs? And that would obviously be right up here. And then we come down and we do a little, my line is not very straight, but it's not at the beginning of expiration, it's not at the end of expiration, it's in the middle of expiration. In fact, at the peak of inspiration and at the end of expiration, the air pressure in the lung is actually equal with whatever the air pressure out here is. Okay, this is a complicated graph, and when I do this face to face, it's just as complicated. I do encourage you to either watch this part again, or even better yet, go to the book and kind of really try to follow it out and reason it out. It should make sense, if not initially, after a couple trials. Okay, so hopefully at this point we have demonstrated that changing the volume of the lungs is going to drive inspiration and expiration. And again, that follows this physics principle where what we say is if you change the volume of a container with air, then you change the pressure. If you change the pressure, you now create a pressure gradient, and air is going to move down its pressure gradient. All right, let's take a look at the role of the pleural membranes in ventilation. So briefly, I'm going to show you this image from the last video. This describes the double pleural membranes. You have an outer and an inner layer, and they are kind of sealed together by serous fluid or pleural fluid. Now let's take a look at this blue line in this graph, and you'll see that the blue line describes the pressure that is between those two membranes. Now, I don't really care about how it oscillates in this graph. What I care that you know is that the pressure in between the two pleural membranes is going to be less than the pressure in the lungs. So we can kind of, in a simplistic way, call that negative pressure. It has a lower pressure than the pressure in the lungs. So what does that actually translate to? Well, what that means is that because there's low pressure, when you have one membrane and another membrane and there's like a low pressure, it sticks them together. Think about the opposite. Think if there was a strip of serous fluid that had a high pressure, what would it do? It would push them apart, right? So by having a low pressure, it sucks them together. Okay, this is advantageous to us because it actually helps us do ventilation. So the outer pleural membrane is attached to the thoracic wall. It's attached to the rib cage and the muscles, and then the bottom layer is attached to the diaphragm, whereas the inner layer is attached to the actual alveolar sacs and all the blood vessels that are complexed with it. So when you expand out your thoracic cage, you pull only on the outer membrane. but because the inner membrane is stuck to it, you pull the inner membrane with it. And by pulling the inner membrane, you pull on the lung tissue. Now, if this seems obvious to you, then let's take a look at a situation where that interpleural pressure, that negative interpleural pressure, gets ruined. And that happens in what is called a collapsed lung or a pneumothorax. So a pneumothorax is going to be where there's some sort of breach in the interpleural space, so that, remember, the pleural membranes have a negative pressure in between them relative to the atmosphere. So if I make a puncture and suddenly open that area up to air, well, air moves down its pressure gradient, right? So air is going to come in and separate those membranes. So the outer membrane will stay attached to the thoracic wall. The inner membrane will still stay attached to the lungs, but there's nothing really keeping them together anymore because the pressure has equalized with the atmosphere. So they separate. And so you can move this one all you want, but this one is not going. And that is what we call a collapsed lung. So it happens when you puncture those pleural membranes, and then the lung actually kind of shrivels up on itself because there's a lot of elastic fibers that are embedded in the lungs that are normally kept taut by that negative intrapleural pressure. And so when you release that pressure, they go, they recoil like typical elastic does, and that lung crunches up. So here's another image. So EMTs sometimes will take calls where someone has a punctured lung, and they've got some pretty nifty technology to basically suck the air out of the intrapleural space to try to pull or re-inflate that lung and get it to re-expand. So what would a collapsed lung be doing in terms of ventilation? Not much, because again, you're expanding your rib cage, but now your lung isn't doing anything with it. So on that note, I want to talk about two terms that are used to describe our lungs' ability to kind of expand and contract. So the term that we want to introduce is compliance. Compliance is the lung's ability to stretch. And you definitely want to write that down. Compliance is the lung's ability to stretch. Where elastance is the lung's ability to recoil. And recoiling lungs are basically driven by elastic fibers that are embedded in the alveoli. So elastance is a property of the elastic fibers. Compliance is more a property of how thin the walls are. So we see like decreased compliance when we get lungs that have a lot of fluid in them, or have a lot of scar tissue. If you've ever seen scar tissue, even on the skin, for instance, it's always thicker and less stretchy than regular skin is. The same is true in the lungs. If you have a lot of scar tissue, then your lungs don't stretch very much, and so they're not very compliant. Having good compliance and good elastance is awesome for your ventilation. When you have some sort of problem with your elastance or your compliance, ventilation becomes difficult and labored, and as we're going to see in a future lecture, that can impact how much gas exchange you have. Okay, so let's take a look at asthma. Most of you probably know of somebody who has asthma if you don't have asthma yourself. It's very common. Asthma is considered to be almost an immune disorder, where something triggers an excess release of histamine into the bronchioles. So remember, the bronchioles are going to lead into the alveoli. Now the bronchioles have a pretty small diameter as it is, but if you constrict that diameter even more, you're going to increase the resistance of that tube and therefore make it more difficult to move air from out here and into your alveoli and back again. In other words, ventilation will become really difficult. So the analogy that I want you to consider is if I give you two straws. One is that little coffee straw, one is the Slurpee straw, and then imagine that you have a nose plug on and you're going to stick one of those straws in your mouth and then make a seal with your lips so that in order to move air, you have to move it through that straw. That's the only way you can breathe, is through that straw. Which straw do you want? The coffee straw or the Slurpee straw? The Slurpee straw, of course, because it's got a bigger diameter and therefore less resistance, it's easier to move air. Likewise, your bronchioles have a certain diameter to them, and if you make that diameter too small, it becomes the coffee straw, and it becomes really difficult to move air down such a narrow passageway. So asthmatics have trouble ventilating when they get an asthmatic attack because their bronchioles constrict. Now, I mentioned histamine. So there are a number of triggers for asthma, but I'm going to use an allergen trigger. So if you have an allergic reaction, like for instance, my husband is an asthmatic, and one of his triggers is rat and mice dander. And that is because he worked with rats and mice for years and years doing pharmaceutical research and had developed allergies to them. But if we have like a rat in our garage and he goes out there and the dander gets kicked up and he inhales it, he can't breathe. I mean, there's just no ventilation, no movement of air. And that is because the allergen triggers the mast cells that line the bronchioles to secrete histamine as part of their inflammatory response. Well, it turns out that the smooth muscle around the bronchioles responds to histamine. And the response is not to relax, but rather to constrict. And it induces bronchoconstriction. So just as a quick comparison, when we learned about histamine before, we learned about it with blood vessels. So histamine causes the smooth muscle around arterioles to relax, but around bronchioles to constrict. And this kind of should make sense. If you're getting an allergen into like some part of your lungs or something pathogenic into some part of your lungs, you don't want to keep letting stuff into that part of the lung, so you try to constrict your airway to prevent it from going into the alveoli where it could cause pneumonia or some other sort of damage. So it's a useful and sensical response of our body to induce bronchoconstriction when histamine is released, but asthmatics overdo it. They oversecrete the histamine, and sometimes for kind of a benign or non-pathogenic reason. And if they oversecrete it, then all of the bronchioles are going to constrict, and when that happens, the resistance goes way up, and consequently, they have a lot of trouble ventilating. Now, in addition, histamine is going to start the inflammatory process. One of the outcomes of the inflammatory process is basically drawing more fluid to that area, and so you can see that here in this image, kind of a double whammy. Not only do you decrease the diameter of your airway, but you also draw fluid out into the airway, further decreasing the airway size, and so together they really increase the resistance, and therefore ventilation becomes really difficult. So you probably are aware of this, that an asthmatic, so you are probably aware that a person suffering from a severe asthma attack can die from it simply because of their inability to ventilate sufficiently. All right, now that we've covered asthma, let's come back and take a look at the surfactant secreting cells. So this is an image that just shows you again the alveoli. And I want to go through a little bit more with the alveoli histology. So remember that the alveoli are mostly type 1 cells, and those type 1 cells are just simple squamous epithelial cells, thin, flat, there to make gas exchange easy. And then we have our type 2, which are our surfactant secreting cells. And then I already mentioned this earlier in the lecture, but it bears repeating. And that is, there's a bunch of elastic fibers in there as well. And the elastic fibers allow that when you stretch out your lungs, they recoil pretty easily. So let's take a look at the role of the type 2 cells. So the type 2 cells are also called surfactant secreting cells because they secrete this thing called surfactant, which is kind of like an oily, soapy mixture that prevents the alveoli from collapsing. So what I want to do first is talk a little bit about water. Well, it turns out that your alveoli have a layer of water on them, so they're moist, right? They're not dry. There's moisture. They're not dry like skin. They're wet. But the alveoli are really small, and water is sticky. Water likes to stick to other things and other water molecules. And so when that happens, when you expire or let air out, the alveoli start kind of coming together, and of course there's water here and water here, and if they get too close they can stick together, and then it becomes really difficult to pull them apart, as we're going to see. So surfactant is being secreted to basically make water less sticky, so that when they come together they're not compelled to seal together by a bunch of hydrogen bonds, because the soaps and other oils get in the way, and then they just kind of stay like this, and then it's a lot easier for them to inflate and deflate and inflate and deflate, and it doesn't take as much work. So I have a video here, a couple videos that will show this. So in this case, I am dropping pure water on wax, and what I'd like you to note is the size of the water drop and how well it comes up off of the wax paper. And now I'm going to take soapy water and make the same size drop right next to the water droplet. And you'll notice that it is, in fact, not sticking together, that the soapy water just kind of runs everywhere. And that is because soap, like oils or surfactant, gets in the way of water molecules hydrogen bonding with other water molecules and being sticky. And so soap and surfactant makes water less sticky. And that's evident here. The soap made the water less sticky, and it just kind of ran everywhere instead of staying together as a nice tight bead. So I also invoked my children into this little science project where I took 2 microscope slides, and one of them is going to add pure water to the 2 microscope slides, and the other is going to add contact lens solution. And they really have no idea what's going to happen. They're just being asked to do this. And I didn't have my mic on, so I'm going to have to narrate what we were actually doing. So you can see Owen with the mask has put pure water on and now put the second microscope slide on. Zachary used contact lens solution, which has oils and surfactants in it. And so you can see what they're doing. So now what they're going to try and do is actually peel them apart and see which one is easier to peel apart. So Owen has the water one, and you can see he's really working on it and finally got him apart, right? And he's like, interesting. Okay, let me try this one. Oh, wow, that separated fast. All right, let me put it together and see if I can peel it apart. And so he is having a little bit of trouble, but he does get it apart. And then I ask him, well, which one's easier? So now he's trying the water one again. He goes, oh yeah, that's really hard. And then Owen's like, yeah, okay, I want to try. So Zachary tries. Zachary peels the water one apart. And he goes, yeah, okay. And now give me the other one with the contact lens solution. And it just falls apart. So he's like, wow, that just came apart really easily. So his last words there, if you saw them, were this one. And that was in response to the question, which one was easier to pull apart? And it's the one with the contact lens solution, because contact lens solution has some like surfactant-like properties to it, makes water less sticky, and therefore it's easier to pull apart. So in this image here, you can see a drawing that describes the importance of surfactant. Up here, we have our alveolar sac, and there's a thin layer of water, and of course that water is kind of pulling towards the other water molecules on the other side, and they really want to stick together. So when you expire and air leaves, the alveoli have a tendency to basically go and stick together and seal, and that is alveolar collapse. But your surfactant secreting cells, again, produce a bunch of oils and other things that interfere with the hydrogen bonding. So when you expire, the alveoli stay open. Okay, so let's talk about COVID-19. COVID-19 infects type 2 surfactant secreting cells. So what do you think that does? By infecting surfactant secreting cells, your surfactant secreting cells stop secreting as much surfactant. Your alveoli have a tendency to collapse. But moreover, we know that when a virus infects a cell, the body's response is to destroy that cell. Type 2 cells do not just secrete surfactant, but they do a bunch of other things too. One of the things that they do is they prevent other bacteria from getting down there. They make it inhospitable to other bacteria. So by destroying your type 2 surfactant secreting cells, you open yourself up to alveoli collapsing, which would cause respiratory distress. You open yourself up to a secondary bacterial infection that has a lot easier time staying down there without the surfactants secreting their antimicrobial material. And they also act as inflammatory cells. They do stimulate inflammation when they sense that something is wrong, like, hey, I'm infected, something's definitely wrong. And they're also the stem cells or the progenitor cells for the type 1 cells. So there's so many things going on that's awful by infecting those particular cells. When type 2 cells become infected, and the body mounts an immune response to destroy them, the entire alveoli basically shreds apart, develops a bunch of material, becomes hardened and proteinaceous, and you lose your ability to reform or regrow your alveoli because they were the progenitor cells or the stem cells that would have made new type 1 cells. So you have this horrific inflammatory response that draws a lot of fluid, causes the pneumonia, you destroy the type 2 cells, the alveoli breaks and collapses on itself, and then you have nothing to repair it. So everything I just described explains why people go into respiratory distress. So again, it's both the alveoli collapsing and the inflammatory response, which people develop pneumonia with. Because again, inflammation draws fluid, so you draw fluid into the airspace. And then you could set yourself up for a secondary bacterial infection down there. And if you try to recover, your recovery is going to be very, very, very slow. Because the way that your lungs would repair themselves is by using the type 2 cells to basically produce new type 1 cells and reconstruct your alveoli. But they're destroyed. So there's nothing there yet. So this is probably a major contributing factor to why we see people who have had COVID-19 have exceptionally slow recovery times. All right, that concludes the lecture. Thank you so much, and I will see you guys on Monday.